Welcome back to Juniata Just Connected for today's Master's Program Monday feature. About a month ago, we were here with Bioinformatics at the Contamination Source Identification Lab, or CSI, with Dr. Lamandella. Good to see you again.
Thanks for having me.
And we're going to learn a little bit more about bioinformatics. And as we're learning, students who are seeking graduate admission, please go to Juniata College graduate programs. Start that application for admission. In fact, application reading for spring admission is already underway, and congrats to all of our admitted students for our spring enrollment term. But with that, again, Juniata College graduate programs, check that out. You could also complete an information request or also set up a Zoom information session or a campus visit as well. But with that, again, we're back to bioinformatics, and here at the CSI lab where Dr. Lamandella, the last time we were here, showed us around. But we've got another space that we're going to check out, along with having the opportunity to meet one of our graduate students, Jillian. So Dr. Lamandella, take it away.
Thanks for having me back, and welcome back. Again, this is where the molecular magic occurs. I'd like to introduce Jillian Leicester, who's one of our graduate students in the bioinformatics program. And Jillian also has the opportunity to work with CSI, our laboratory here, full-time. And she has really been part of helping us develop this revolutionary infectious disease diagnostic technique. What you can see Jill's doing right now is combining a series of clinical specimens that we are going to subject to high-throughput DNA sequencing. So something that you get to do here in this bioinformatics program are hands-on biotechnology techniques, one of which is this revolutionary DNA sequencing. So right behind Jason here is our Illumina NextSeq 550. So DNA sequencing has really changed dramatically over the past 10 years. So we've gone from, like when I was back in graduate school, you could only generate one DNA sequence at a time. And remember, you might remember from before, that DNA sequence is our thumbprint for what pathogens are in our sample. Now, with this modern day technology, we can do up to 800 million DNA sequences at one time. So we can parallelize a lot of different clinical specimens at one time and try to get those results back to physicians really quickly. And something we've focused on, and something Jillian and some of the other graduate students are working on, is our patent pending technology to do this. So we have two infectious diseases that we're focusing on. One is Tick-Borne Diseases. So many of you have probably heard of Lyme's disease. Devastates many Americans each year. In fact, Pennsylvania is the #1 state for new Lyme's disease cases each year. And so we've created a panel that will detect over 100 different tick-borne pathogens, whether they're viruses, eukaryotic organisms, bacteria, and we do all that on the robotics that you saw last time, as well as the DNA sequencing. So once those samples come off the robot, what Jill's doing is combining them right now, and we're going to load them onto this flow cell. So this little flow cell, believe it or not, is worth about $4,000. So the sequencer automatically, all the fluidics and everything are controlled inside of here, and it's going to inject the samples that Jill's preparing onto this flow cell. So the DNA is then going to hybridize over this entire glass flow cell. And then it's just what we call a sequencing by synthesis reaction. We literally build the other strand of DNA and decode the sequences based on what color fluorophore is being added. And so then all that data that gets generated here then gets piped to our bioinformatics pipeline, where we compare it to databases that we have in-house that have over 10,000 different pathogens. And so right now we're doing a lot of the heavy work. A lot of our students are helping with the heavy lifting in terms of our clinical validation. So we're taking literally this technology from the bench, right, to physicians. And one of the biggest things we have to do is make sure it works in humans. And so we're in that clinical validation step, and it's been really great to partner with our students to develop this technology.
That's great. Can we get a close-up just to see how Jillian is loading right now currently?
Got a very steady hand.
And about how long does it take them to load and get it prepared for transfer?
So this by hand step, this is the only step we don't do robotically. This step will take about 10 or 15 minutes. So she's combining all 96 samples that have a unique barcode on them so that we can tell them apart later. So once she's done with that, then that gets loaded with this flow cell right onto the sequencer. And so based on the data that come off of this sequencer, then we can identify, okay, which pathogen is causing this different disease state. So in other words, is it a Borrelia that is causing this individual to have Lyme disease? Is it some sort of virus that's causing this person to have some other type of tick-borne disease?
Yeah, excellent. Well, appreciate you showing us more of the CSI lab. Jillian, keep up the good work, and all students tuning in. Again, Juniata College graduate programs, go complete your application for bioinformatics and check out all of our other graduate programs too in the MBA, Masters of Organizational Leadership, Masters in Accounting, and Data Science. Tune in again for our next Masters program Monday, but until then, we'll see you soon.